Greetings. This is a recreation of a talk that I gave at the APS March meeting and the DPG Spring meeting in 2024. It's also a summary of the paper linked in the description. This work focuses on the fission of membrane tubes, specifically double membrane tubes. During the process of mitochondrial fission, for example, summarized here, the two daughter organelles are connected by a tube that has to undergo fission. In mitochondria and other organelles with double membranes, this is a double tube. Real fission events are catalyzed by dynamin, which is a protein which wraps around the tube and squeezes it to catalyze fission. I'm going to ignore the role of dynamin for now, focusing on the optimal path for membrane fission for just the membrane. Further work will explore how mechanical stress alters this optimal behavior. This work utilizes self-consistent field theory. For a more in-depth description of the method, I invite you to watch the other videos on this channel. But in summary, we consider the state of a system as described by a set of monomer concentrations phi, where gamma is an index identifying the monomer type. This concentration profile gives rise to a set of chemical potentials. These are the potentials felt by a monomer of type gamma at position R. These potentials in turn give rise to probability distributions of monomers. To calculate these distributions, taking into account chain connectivity, I model the lipids as Gaussian chains and use standard techniques described elsewhere on this channel to calculate their statistics. The average concentration can be calculated from the probability distributions. If the initial and final concentrations match, then the system's happy, i.e. it's in a stable or metastable state. Otherwise, the difference can be used to calculate local exchange chemical potentials which drive the evolution of the system. This method produces all of the structures that one might expect: planar membranes, tubes, multi-layer tubes, micelles, worm-like micelles, and other structures not shown here. But I'm not just interested in stable or metastable states. I want to know how one structure turns into another structure. Without a nice reaction coordinate, I need something more general. I can write the free energy of the system as a function of its configuration in some huge space of possible configurations. If I think of the concentration at each point in space as a variable describing the state of the system, then the state space has as many dimensions as there are points in my system. In practice, I represent the system with order 1,000,000 points. To illustrate the method, however, here's a sketch of a free energy landscape in just 2 dimensions. A system starts at one point on the landscape with a given free energy, and it evolves by descending the free energy curve until it reaches a minimum. But I don't just want a local minimum. I want to know how to get from, say, this minimum to this one. This involves creating a set of replicas forming a path or string through configuration space. The whole string is relaxed simultaneously under the constraint that the points remain equidistant in configuration space. We can look at the fission of a single tube. It starts with a tube and runs like this. Going over that step by step, it first constricts locally, and then the constricted region collapses. The tube is now blocked, and the water within it cannot flow. The collapsed part relaxes. If the tube's at a high tension, the collapsed part extends into a worm-like micelle, whereas if it's at a low tension, it can just fission here. In this calculation, it extended. At some point, the constriction point constricts further and breaks off. The tube is now fissioned, and given that the tube and the worm-like micelle are under tension, they retract. The string method doesn't just give me a path, it also gives me free energies all along the path. The free energy first increases as the tube constricts locally. This first maximum is the local collapse, the second maximum is the fission of the worm-like micelle. I'm collaborating with researchers at the University of the Basque Country, led by Anna Chernova. She does cryo-EM experiments on membrane structures. I'll give you some more details about how she does them shortly, but for now I just want to point out that when she looks at tubes undergoing fission, she sees the main structures that I find in my model. As a theorist, it's always nice when experiments find what you predict. But I told you from the beginning that I'm interested in double tubes. I can redo the calculation that I showed you for a single tube, but with another tube around it, i.e. looking at the fission of a double tube. It looks like this, but when I calculate the free energy barrier, it ends up being basically the same. A little lower, but barely. This is likely due to a small amount of pressure from the outer tube squeezing the inner tube. But based on this, I would expect the single and double tubes to fission at about the same rate. So now let's look at what Anna sees. First, I have to tell you a bit about her experiments. She sets up a system containing single and double membrane tubes and flows them past an array of micropillars. They stick to the pillars and have a tension that's related to the flow rate and the stickiness of the micropillars. Rather than embarrass myself trying to explain her experiments, I'll refer you to the paper for details. The point is that she gets single and double membrane tubes that she can either watch dynamically using fluorescence microscopy, or freeze an image using cryo-EM. Fluorescence gives dynamical information, but doesn't have the spatial resolution to see the details of the membrane rearrangements. Cryo-EM can show the membrane details, but just gives snapshots. It's also really difficult to capture rare events because, well, they're rare, and to see them you have to happen to freeze at just the right moment. In these fluorescence microscopy pictures, you can see the double membrane tube. You know that it's a double membrane tube because of the high light intensity. The membrane fluoresces, so where there's more membrane, there's more fluorescence. The inner tube fissions here, and then it retracts. In addition to just seeing it fission, she can extract the fission rates. She finds that the inner tube of a double tube fissions more easily than an equivalent single tube. If you've been paying attention, this might seem strange, because I just told you that the free energy barrier should be about the same regardless of whether there's a tube wrapping around the inner tube. The resolution to this little problem comes from the fact that double tubes can do things that single tubes can't. For example, they can fuse together, as shown here in SCFT and also in cryo-EM. When I calculate a string that includes this possibility, here's one example of a thing that I get. There are also other variations, but I want to highlight this one. The inner and outer tubes first bend towards each other. They then connect, forming this stalk or hydrophobic bridge. The inner tube then constricts, but it doesn't have to constrict all the way. Instead, it forms a pore. The pore is stabilized by the presence of the stalk, and it first closes on one side, and then the other side. This gives rise to a structure similar to what we saw earlier, but with a stalk connecting the collapsed inner tube to the outer tube. The stalk then breaks off, and the connection between the two capped-off tubes breaks, allowing them to retract. I'll play it one more time so you can have a closer look. As before, I want to look at the likelihood that this pathway will occur by looking at its free energy. Here I show the free energy along the simple pathway in red, and the more complicated fission by fusion pathway in blue. The total barrier for both pathways are similar, though the barrier to the fission by fusion pathway looks a little lower. It looks like this new pathway is preferable and occurs faster, but only by a little bit. But even if the difference in barrier is negligible, the presence of a second independent pathway means that the system has two different independent ways of undergoing fission. Even if the two barriers are the same, having a second pathway should speed up the fission process. You can think of this as having two different directions in configuration space that the system can fluctuate in order to undergo fission. In addition, there's this shoulder where a stalk forms. In this curve, it looks like the free energy increases monotonically, but the other calculations we have suggest that the stalk is actually weakly metastable. The stalk intermediate might therefore act as a jumping-off point, helping the system to jump over the barrier. So in this work, we've used self-consistent field theory to model changes in geometry and topology of biological membranes, specifically tubular membranes. A single membrane tube undergoes fission by a simple leakless pathway. Experiments indicate that even at the same tension, the outer tube assists the fission of an inner tube in a double membrane system. This can be explained by the discovery of a new pathway, whereby transient fusion events between the membranes can catalyze the fission of the inner tube. Thanks for listening. If you're interested in this work, I invite you to read the paper. It's open access, so it's pretty easy to get your hands on. Throughout this video, I've included references to a number of different methods without going into much detail about the methods themselves. If you're interested, I have some videos about SCFT as well as the string method, and I'll add links in the description. The paper also gives details about the specific implementation of each method in this work.